Hello everybody, welcome to this new section of Lallemand Animal Nutrition Animal Companion webinar. I will be pleased to share with you those 40 minutes and I will present you technical information about new yeast derivative and hydrolyzed yeast that may help you in your companion animal feed strategy. First, when it comes to this yeast derivative, first, it's really integrating the global context of finding new alternative source of protein. And in Lallemand R&D team, they dedicated specific time looking for specific available biomass that can be transformed into a nice nutrient specific functional solution that can be in two hands providing health but in the same time being a natural ingredient and sustainable solution for companion animal nutrition. Doing this, many of those yeast-based ingredients are promising solutions. The global family of those yeast derivatives are quite big. We have two main categories. The first one is yeast fraction. So as a fraction, we have two main categories, which are yeast cell walls and yeast extract. And we can easily understand that it's coming from an extraction of a whole yeast cell. This family of whole yeast cells include those wonderful live yeast that keep alive and use as a probiotic. And we have a lot of different categories, subcategories with inactivated yeast. That can be the first one, the first level, inactivated yeast directly, hydrolyzed yeast coming from endogenous enzymatic process. The one we will speak a lot today is hydrolyzed yeast, very specific, and because it's coming from a specific exogenous enzymatic process, kind of unique product. And the last one is inactivated yeast but enriched yeast, like for example, selenium. So in those categories, of course, we don't have the same property, the same functionality. And the simplest one, the inactivated yeast, okay, the CP content is nice, but in front of this, the digestibility is not pretty good. And functionalities are quite low. And that's the main limit of the product. When it comes to the second step, the hydrolyzed yeast, then it really depends on first the quality of the inactivation and then what are the endogenous enzymes that remain that lead to a problem of usual consistency. The robustness of the product sometimes may cause some problem. In yeast extracts, it's quite different because here, of course, we don't have the wall. We are mostly what is intracellular content, meaning that these soluble proteins are highly concentrated. It's not the same property. It's a highly digestible. But we face in this case two main problems. The first one is the cost, the price, and the second one is the availability, especially for our sectors. And the one we want to spend time today on is hydrolyzed yeast. In this category, we are using this specific biomass, and that's where the sustainability is coming from because we have a lot of this biomass. And from this biomass, we are able to apply a very specific inactivation, soft one. And then we use mostly exogenous enzyme, chosen one, coming from a a protein engineering choice in order to obtain targeted product. So in this case, we may have also different quality of hydrolyzed yeast, but most of the time the digestibility is improved. And doing so, we are also helping a higher expression of functionality, because we are not speaking about the same product. We have an initial biomass and we create something really different. It helps to have a higher global digestibility. Because the process is stronger, it's also a way to improve consistency, robustness, and of course, availability, and the price is an important point to consider. So in the process, because this kind of product is coming from the industry, it's coming from a process. Inactivated yeast, very simple, coming from an inactivation, sometimes too strong, that is also destroying some quality of protein. Hydrolyzed yeast, we have a first step of inactivation, then using endogenous enzyme, we have a first step of degradation, as I said, sometimes irregular. And the one we will focus on today, hydrolyzed yeast. So depending on the choices, depending on the quality of the factory, we can have several hydrolyses on the same biomass, leading to a very specific and unique product at the end. So much more complex process, but leading to a higher value for the solution at the end. What is this biomass? So a yeast is a very specific element. The yeast has a very complex wall, especially when it comes from bioethanol. In order to protect from the medium, the yeast will develop a specific wall, inside which we will find glucan, mannan, glycoprotein, and 2% of chitin usually. And inside the cell, the composition is so rich. We have some protein, big one, some, let's say, peptide, medium size, a bit of amino acid, but not that much. But we have also nucleic acid, enzyme, cofactor, vitamin, a lot of components, and then you can already understand that depending on the process we will organize, we may cut some of those, keep some others, and that's how and where all the R&D ability and know-how was able to express. So thanks to the process hydrolysis from a source of protein, then we will be able to create a higher digestibility, a release of small free amino acid and small oligopeptide. But in the same time, coming from this very specific wall, we will have a very original functional dietary fiber. And beside this, all the vitamin, trace mineral, very specific, highly bioavailable, will be also protected from the process. And then, coming from a well-controlled, standardized hydrolysis, we may have such a specific product to offer. That's where Lallemand Animal Nutrition has made all the choices that lead to YELA-ProSecure. One specific biomass. And when I say specific, coming from bioethanol, so very specific yeast strain and expression of this strain. One brand new specific plant in which we apply the process that we decided, that we were able to create in order to have the targeted product. And at the end, one specific and I should add unique hydrolyzed yeast. So to speak about the composition of the Yela Prosecure, we have three main parts in this product. The more valuable one, the crude protein. We are above 42% of total crude protein in the Yela Prosecure, inside which we have a high quantity, a high portion of free amino acid ready to be digested, inside which we have small oligopeptide. The same, most of them will be able to be digested without no digestion. It's a direct absorption. And we have peptide, medium peptide. The global digestibility is close to 95%, which is very interesting for a yeast derivative. The second fraction is the so-called dietary fiber, as you understood, mostly coming from the wall and very specific wall of those yeast that produced ethanol. The total dietary fiber content is around 40%. Using the Prosky method, AOSE and 985.29, the soluble dietary fiber is 7%. The insoluble dietary fiber fraction 32%, with no inert fraction, almost nothing inside this fiber fraction will not be fermented. And the last part, but still valuable part, will contain vitamin, trace mineral, trace element that will also be interesting for our companion animal. Those are bioavailable nutrients. If we focus now sometime on the crude protein fraction. So again, we are speaking about hydrolysis, and not one but several hydrolysis on this biomass. So from big molecule, we are able to target exactly the modification of those big molecule to small compounds. Free amino acid, very small peptide, small peptide. That means we cannot compare the initial biomass and this unique hydrolyzed yeast. We are bringing new, first, digestibility, but thanks to this type of digestibility, new property will come, new functionalities will come. How to describe the efficiency of the hydrolysis? First, describing the size. In the soluble fraction of Yela Prosecure, we have more than 55% of the molecules that are below 307 Dalton, meaning the smallest size possible when it comes to nitrogen content. It is free amino acid that can be absorbed again without digestion. We have oligopeptide, still very small molecule, 12%. We have small protein below 6,000 Dalton. It's not big. And we have keep some not big but medium size protein with 6%, and mostly related to the walls of the Yela Prosecure. And you can understand that the intestinal assimilation will be very easy, direct, and that will create the functional effect that we will describe later on. Those amino acids, the one in the free form, but also the global one. Luckily, with Yela Prosecure, we have more than 18 different amino acids. And also, luckily, the composition is very interesting because we are very close to the ideal protein composition with a high level of secondary branch chain amino acid. Those ones that are very valuable, not easy to find on the market, and that also related to many functionalities. Usually for young animal, but not only, but also for old animal, the protein digestion is not easy. Depending on the type of protein, it may be very long. And it also may be in a bad yield of digestibility. It's a complex and long process, this digestibility of protein. And the stomach is very important in this process. And sometimes we are facing some lack of efficiency of this stomach. Protein digestibility, which is important. I will not say that it is not important, but it is not expressing what is becoming more and more important for nutritionists in many species is the kinetic of absorption. Inside this digestibility, it's quite important to be able to describe how fast is this digestibility. And you can understand that depending if it's a flash, fast, in big amount digestibility compared to a very slow step-by-step digestibility, the functionality and the release of amino acid will be different. This kinetic is now known as a way to express very specific functionality. Not easy to describe. We have a standard method coming from human, coming from food. It's a biocene method that is quite interesting to describe what is flash, very fast, right after the stomach, fast, slow, but even resistant digestibility, resistant protein that will not be used by the animal, but probably be used by pathogen. This biocene method, because I will show you some result coming from this method. In this method, we are mimicking what is happening inside. So it's an in vitro model, but very efficient in vitro model that will mimic oral ingestion, the stomach phase, and the global intestinal phase. And then you can control all parameters, and then you are able to check how one component, how one diet, one raw material may react through this model. Of course, we apply it to LEBRETON. First, and it's good to know, from one batch to another one, we are very regular. The second one, which is very interesting, is you see that more than 84% of the protein will be absorbed right after the stomach. 84% of the protein will be directly digested, absorbed by the animal. What does it mean? It's a very small portion of this protein content in the yellow procedure that will still in this small intestine. And it's a big release in a small period of time. Another way to see that this model is a good one, that we see that after 4 hours, we are going to the known, let's say, digestibility of our product. Just to make things maybe more clear, in your choice, the choice you will make in your feed strategy, in your food strategy, the component, the raw material will not react the same. They may have the same global digestibility, but with a difference in the kinetic. I will not say the worst, but an example of a slow kinetic of digestion for protein is soybean meal. It will take hours before releasing the global amino acid. And the opposite, synthetic amino acid, will be a flash release. All of them in a very short period of time will be absorbed. The same for most of the Yellow Prosecure. You remember, 84% of the protein of Yellow Prosecure will be absorbed in very short period of time. And of course, we need to understand that in the animal, it will not be an individual, let's say, process of absorption. It's a complex, and it's an addition of all the fraction coming from all the raw material. What that means is global peak of absorption right after the stomach will be accumulation coming from synthetic amino acid if you use it, Yellow Prosecure if you use it, plasma if you use it. For this kind of product, you will have accumulation of those free amino acid and very small oligopeptide. The first functionality that will be directly related to this content of free amino acid and this fast absorption in a nice quantity and in a balanced quality of amino acid is gut health. And when we say gut health, it's firstly, it's a intestinal mucosa that will have the first positive impact of those, especially secondary branched-chain amino acid. Speaking about the surface of absorption, the shape, the tight junction, the gut barrier expression will be much better thanks to this fast and full covering of the requirement of those epithelium cells. So very positive. Of course, an impact on the gut microbiota, because as soon as you absorb very fastly the protein, you have less protein available for proteolytic bacteria, and it's one way to avoid abiosis, the opportunistic bacteria development that we are not looking for, of course. And coming from this, gut immunity, antioxidation will be also helped by all of those. We should spend at least 1 hour on this one. We will spend 2 minutes. Taste, flavor. Amino acids are mostly related to those very specific components of taste that will be able to modify the feed behavior. That's something very important, especially when it comes to companion animals. In relation to the quality of amino acid, but I will insist especially on the kinetic of absorption of a nice and balanced quantity of amino acid, and this is depending on type of animal. I will take the example of umami. For the human, 2 amino acids are sufficient to express the umami taste coming from specific receptors. When it goes to, for example, swine, it will be 7. For dogs, it will be different. So every mammalian will express the umami, let's say, taste, but with different receptors and different sensitivity to different amino acids. But again, it's a matter of amino acid fast release. Of course, when it comes to amino acid, it plays on skin appendages. When we speak about hair, it's almost protein. It is almost composed of protein. And cysteine and methionine, so sulfur amino acid, are particularly important to the quality of those hair fibers. Every time we are facing an immunodepression, every time we are facing a specific metabolic time stress, requirement may be modified. The proteic requirement and specific amino acid requirement will also be modified. When we look at the pigmentation, the intensity of the black, tyrosine and phenylalanine will also play a specific role. Tyrosine, tryptophan, cysteine, looking at the skin and the pigmentation of the skin, they will be also involved. So there are many reasons to take into account quantitative position of those amino acids, qualitative with the kinetic of absorption. So as a sub-conclusion of this first part about the protein, yes, with the Yella Prosecure hydrolyzed yeast, you will face a flash digestibility. That will be part of your specific strategy that will include all your choices that will bring some protein, but this Yella Prosecure will be very fastly absorbed. You will have a peak effect of a nice composition of amino acids. It will help to protect the enteral mucosa. So this enteral nutrition will be done with all the benefit that we can clearly think about. The umami, thanks to the glutamate, but not only the glutamate, but also the aspartic acid, those one will make a difference. And I will show you some trial later on showing this difference in the choice, but also the feed behavior of the animal, leading to a better appetite. And the last, but also very important, skin appendages. The way that when you use the Prosecure, it will contribute to a better, let's say, hair skin expression. Just to have a look to the second fraction, which is a functional dietary fiber. If you give a sample of Yella Prosecure to a lab, it will come back with what you have on the screen. A global dietary fiber close to 40%. And inside this global dietary fiber, which is the main and the best way to describe the whole composition in fiber of your product, we are quite lucky with a control and quite low soluble dietary fiber fraction, and an insoluble dietary fiber fraction, which is mostly fermentable because the inert fraction is very, very, very low. In the choice that you will make in your food, in your companion animal food, this quality of dietary fiber will be quite unique because those components you will not find it in other raw materials. They are quite specific. And being specific, also one word saying that there is no anti-nutritional factors. And at the same time, we have no contamination and no mycotoxin, which can happen with classical, let's say, fiber, raw materials that will provide fiber. And the insolubility of those components are mostly due to small amount of chitin that will create with glucan an insoluble expression. That's why it is mostly fermentable due to this chitin link. So with Yella Prosecure, we have those two main components. The first one, physical impact, wall stimulation, gastric emptying. So we are able to modify the gastric emptying, water banding, water holding capacity, a bit an impact on the viscosity, and a bulk effect. Small one, depending on the level of inclusion. And on the other hand, a bit more complex, the physiological impact. Okay, those fibers will not be digested. They will be used by some specific bacteria inside the microbiota, and they will be processed by very weak bacteria, anaerobic Ruminococcus fibrobacter, and then some other bacteria will be able to use the first metabolite to create short-chain fatty acid. Here we are also playing with the kinetic. The time needed to have this cross metabolic feeding, it's a long. First, it must go to the colon, and then inside this, it's a long process of fermentation. So the release of those short-chain fatty acid will be quite late after the meal, which is also something interesting. And then it will also be a substrate for many positive bacteria. Most of them are very weak when it comes to fibrobacteria. Immunococcus, they are very, very weak. It's totally the opposite of opportunistic, but feeding them is one way to contribute to a kind of protection. You feed them, they will be able to develop, and doing so, they will change the pH, they will create a new diversity in this microbiota, which will be positive. I will show you one trial that has been done internally, showing that this fiber fraction of Yellow Prosecure is really active on the gut microbiota. So it's done in vitro. We use a pool of feces and we compare the Yellow Prosecure global one, the insoluble fraction of the Yellow Prosecure, which is really showing what will go into the intestine, and we compare it to inulin. Inulin is a very well-known one and is also one way to check if our model was accurate enough. So in the coming slide, I will show you several colors. Inulin in green, and you will follow Yellow Prosecure insoluble fraction in red compared to a control. So you can see here, insoluble fraction of Yellow Prosecure in red. We still have the full Yellow Prosecure in blue, and you can understand that it will never happen in the true life because the protein fraction will be absorbed very fastly, so we will never have this opportunity to see the full Yellow Prosecure in the colon, but we kept it, the inulin and the control. What is happening in this model? Very nice. First, we see that it's a long-term process. You see the time here, so it's not done first hour after the meal, let's say. It's a long-term process. The concentration, the production of total fatty acid, volatile fatty acid, is very nice with inulin, with the insoluble fraction of Yellow Prosecure, but also with Yellow Prosecure, and very different from the control. When it comes to specific short-chain fatty acid, that's something very interesting. It's like in the book, with inulin, we are looking for this production of acetate and butyrate. So we found it back, showing again that the model is very efficient. It's very helpful. But look at the Yellow Prosecure. The insoluble fraction of Yellow Prosecure, which is very close to the reality, so without the protein fraction mostly, you see that the acetate production, propionate production, but I will insist on the butyrate production, is very nice. Showing that this Yellow Prosecure is used by the specific bacteria, fermented, but with a very nice orientation in the end product, because we have this nice production of butyrate. That's something very valuable, speaking about the epithelium cells development, speaking about transporter, also efficiency. So it's something very nice. If we look at what happened on the main family of bacteria, you see that the global total anaerobes are developed. Okay, we are kind of pushing this diversity again as a substrate for a very weak population. We are helping for a development of those weak population. Better diversity is always something better for the animal. The same for facultative anaerobe, but when you look at lactobacillus, we are also pushing the lactobacillus. Okay, so we are helping the total anaerobes. We are also developing the lactobacillus. It's a kind of boosting effect of Yella procedure. What does it mean? Thanks to this specific composition in fiber, dietary fiber, it's a perfect substrate for weak bacteria, anaerobic, the one we want to develop. It's a really adapted fiber source for sensitive companion animals. And it's part of a global strategy in fiber. What visible sign can be expected from this contribution? Of course, the smell of the feces. In some cases, also avoid belly pain, feces presentation. But at the end, because of those, satiety, but also behavior of your animal. At the beginning, we spoke about three main parts. We saw the protein, we saw the fiber. Now a few words about the third fraction, inside which we have a quite nice content in group B vitamins. It's not nothing. And inside those one, we have 5 vitamins coming from the group B with a high concentration, a nice concentration that will lead to a nice contribution coming from the Yella procedure. And if you look at the B1, B2, the choline B4, but also the B9, that's something that will be valuable. And don't forget, and you know it, that for example, riboflavin is one way to avoid flaky skin. Also, hair loss. Biotin is very well known to also modify pigmentation of the hair. Pantothenic acid is also playing a role in the loss of hair and pigmentation also. That's something that will be visible from the use of the Yella procedure. We are coming to the end of the presentation. I will give you some example of trial done with Yella procedure. It's a new product, but we already have some nice trial. The first one I want to show you has been done on dogs. In the state, Illinois, 20 dogs, and we make a comparison between two types of yeast derivatives, an autolyzed yeast, 1% of inclusion, compared to 1% of our Yella procedure. Same diet, we provide 40 grams of each, let's say, treatment per dog per day, and we check the feed intake of those animals. As you can see, there is a big difference. It's very clear. You see, if you compare the brown and the green one, you see that from the beginning, from day one, we see a difference in the Yella procedure, let's say, intake. The dog appreciate, the dog eat better. And during those 4 days, every day, Yella procedure was at a higher level, and globally speaking, we double the quantity eaten thanks to 1% of Yella procedure. 1% of Yella procedure. You can make a relation with what we say, speaking about the protein content. In yellow prosecure, we are close to 5% total glutamic acid. This glutamic acid, not alone, aspartic acid is also involved, will help to create this, let's say, feed behavior, this appetite. Another trial, again, done on dogs, but this time 40 adult medium-sized dogs. And we make another comparison, this time not with an unauthorized yeast, but with digest, premium digest that has been done on the co-team on the feed, compared to 1% of yellow prosecure also incorporate in the cow team. Same, we want to compare the consumption, but this time the time allowed to eat was half an hour. If you look at the result, it is also something quite clear. This digest is known to be positive and to be good for the, let's say, attractivity. And if you look globally speaking, the global consumption is again quite different. Here we are again speaking about 4 days of comparison. During those 4 days, in average, the difference is big. The difference is big, and you can look at the diversity, it's also notable. And if you look day after day evolution, we saw the average. And if we look at the day after day evolution, the control with the digest, you see that the consumption is decreasing. That's also something to note. When the yellow prosecure, we keep the same high good level. What does it mean? During those 30 minutes, the dogs with the yellow prosecure treatment, they eat more and they eat faster. Again, something positive and statistically, let's say, correlated. Total different trial, this time on horses and specific racing horses done in France. And looking at, okay, we have this kind of comparison with or without yellow prosecure, and this time, what is the body development? It has been done in France. It has been done with almost 50 horses, so it's a big trial, not that usual, and we have 2 main groups depending on horses. One group is done with trotters, and one group is with gallopers. And inside those groups, 2 groups, and with and without the yellow prosecure, we give 45 grams per day. And during a very specific training, intensive training, very intensive training, I should say. And we check the body weight. Everything was under control, and you have on your screen, you are able to see, you have the yellow prosecure in green and the control in gray. So first, you see that there is a difference. During all weeks, yellow prosecure was above in trotter, but also in gallopers. But more than this, okay, the answer was better, but this answer was slightly better. We are speaking about, if we speak about trotters, 4% with the yellow prosecure against 2.1% in the control group with trotter. So that's almost 2% of difference in the animal body weight. The galloper group with a longer treatment duration, 8 weeks, that's something a bit different, because in this case, we have in the yellow prosecure group, we improve the weight, the global weight of the animal, 3.7%. When the others, they lost the weight. It's a minus 0.6%. So it's total difference in the expression. And I forget to say that at the beginning, of course, there were no difference statistically significantly different at the beginning. Here we are speaking about the body weight, but what was the composition of this evolution in body weight? The same trial, but this time the muscle weight, the total fat tissue weight have been checked in the same time that they did a body condition score. Again, if we look at the trotter, so during 4 weeks, look at the difference. With the Lallemand, and again 45 grams per day, we have a difference in the muscle deposit of 1.3% during this training. We have a difference in the total fat tissue weight deposit of 13% when the body condition score improve of more than 4.5%. That's something very visible. That's a first explanation of the body weight evolution. Look at the gallopers. So again, a longer experiment, and look what happened in the control. In this such an intensive training, they were losing muscle. They were losing fat. While with the Lallemand, they were keeping it. But the difference is big. And what is interesting, that we have the same trend for the muscle. The difference between the control and the Lallemand is more than 1.3%. And the same for fat. Look at the difference in the fat deposit, or the fat tissue weight is 6%. That's also one way to show that something is happening on the kinetic of digestion of protein. When you provide this protein after the intensive training, the quality of your protein, of course in relation to the carbohydrate strategy, will modify the way the horse will be able to depose or not, to keep a good, let's say, physical condition or not. And small amount may create big and nice differences. We're coming to the end of this presentation. As a kind of conclusion and take-home message, when we speak about this Lallemand, the main word is kinetic. And when we speak about protein, you understood, we are speaking about a very fast release of a nice and well-balanced amino acid quality. We are hydrolyzing global biomass, creating small peptide, free amino acid, and a fast absorption. Thanks to this fast absorption, we are creating functional impact, mucosal nutrition, gut health, globally speaking, feed intake, feed behavior, something also very important. And I'm not speaking here about the skin appendages that, of course, are also related to the amino acid composition and this kinetic of absorption. That's in one hand. In the other hand, again speaking about kinetic, but this time late release, specific dietary fiber that will be used by specific bacteria to create nice short chain fatty acid. Remember the butyrate. That will help to provide a late release of, let's say, energy. That will play on the microbiome diversity, very positive. That will lead also a decrease in proteolytic activity. Remember, less smell, for example, less smell in the feces. So, thanks to this Lallemand, it's a sustainable let's say, component that may have two different kinetic applied to the companion animal. It's a extreme valorization, and we are quite happy with this valorization. We use a substrate that we may find in big quantity. That's where the sustainability may be found. We create from it a very specific, let's say, solution, substrate, using everything that Lallemand Animal Nutrition know. Functional solution, but also quality requirement, okay, regularity, and from batch to batches, being able to have exactly the same profile of product. It's a new tool that may help you in your strategy to achieve modification in the feed behavior, well-being of the animal, gut health, and with everything related to this gut health, and visible on the skin, on the hair. Really a solution that may help you in the future. Thanks a lot for your attention, and see you next time.